Hi, my name is Kimberly Henderson, and I'm an undergraduate studying neuroscience and cognitive science. I work in Dr. Ralph Ragozzi's neurophysiology lab, which focuses on the effects of nicotine on the respiratory system. More specifically, the lab conducts research to understand how developmental nicotine exposure affects the respiratory system and how this exposure may lead to sudden infant death syndrome. My role in the lab is to determine whether or not there are nicotinic acetylcholine receptors on phrenic motor neurons using neural electrophysiology. By this point, you probably already have a few questions in mind, such as what is sudden infant death syndrome, what is developmental nicotine exposure, what are nicotinic acetylcholine receptors, and what is neural electrophysiology. To start off, sudden infant death syndrome is the unexplained death of a seemingly healthy baby. It usually happens during sleep to babies that are less than a year old. Approximately 2,500 babies die of SIDS each year in the U.S., and it is currently the third leading cause of infant mortality. One possible explanation for sudden infant death syndrome could be defects in the portion of the infant's brainstem that controls breathing. One reason for these defects could be brought on by developmental nicotine exposure. Developmental nicotine exposure simply means that a baby, or in my lab's case, a rat neonate, is exposed to nicotine in utero and later via breast milk. As mentioned before, my experiment is performed to determine whether or not there are nicotinic acetylcholine receptors on phrenic motor neurons. Phrenic motor neurons are just motor neurons located in the nerves leading and coming from the diaphragm. Nicotinic acetylcholine receptors are a type of receptor that bind the neurotransmitter acetylcholine, and this receptor can also bind nicotine. So, how exactly do receptors work? Think of a receptor as a lock, and the neurotransmitter or chemical, in this case acetylcholine and nicotine, as the key. When nicotine binds to these receptors, it causes the activation or inhibition of the neuron, or cell that it is embedded into. Therefore, they are vitally important for neuronal communication. To determine whether these receptors are on phrenic motor neurons, I use 1 to 5 day old neonatal rats with no developmental nicotine exposure and perform the experiment using a neural electrophysiological technique, which basically means I perform a dissection to record the electrical activity of the tissue. In my case, I dissect the brainstem spinal cord out and place it in a split bath of oxygenated artificial cerebral spinal fluid. I then separate the bath in two so that I can add nicotine to the spinal cord without affecting the brainstem. I latch onto a C3 to C5 nerve and then view the changes in the electrical output to see if there are any specific increases or decreases. So far, there have been significant changes that indicate the possibility that these receptors are located on phrenic motor neurons. My research project shows that nicotine does have an effect on phrenic motor neurons in some direct or indirect way. Furthermore, it shows that nicotine has a great effect on the respiratory system. Many questions have unraveled throughout the process, and many more will arise with time and further experimentation. Some of these lingering questions are: what type and subtypes of nicotinic acetylcholine receptors are there on phrenic motor neurons? What would happen if antagonists are added? And do these receptors play a distinctive role in sudden infant death syndrome?